Hi everyone, I'm Lily, and I'll be sharing some of my dissertation research with you today on behavioral flexibility in wild spotted hyenas. Behavioral flexibility is the ability to adaptively modify a behavior when faced with change or novelty. For example, these macaques learned how to wash potatoes before eating them, and these blue tits learned how to exploit a novel food resource by opening milk bottles. One question that researchers are interested in is, why does this kind of high behavioral flexibility evolve? There are currently two leading hypotheses for why behavioral flexibility evolved that suggest that either social or ecological demands were the main drivers of behavioral flexibility. The social intelligence hypothesis suggests that life in socially complex groups, such as those found in primates, selects for high behavioral flexibility. And support for the social intelligence hypothesis largely comes from work correlating brain size with group size in primates. And while brain size isn't the most direct way of measuring behavioral flexibility, historically it's one of the most common. Next, the cognitive buffer hypothesis suggests that environmental variability selects for high behavioral flexibility. And support for the cognitive buffer hypothesis largely comes from work correlating brain size with environmental variability across different bird species and bird taxa. So, in order to understand when or where behavioral flexibility is adaptive, I examine the social and ecological correlates of intraspecific variation in behavioral flexibility using two different cognitive apparatus with wild, free-ranging spotted hyenas. Like Cercopithecine primates, spotted hyenas live in large groups with multiple unrelated individuals and strict linear dominance hierarchies. These are aspects of primate society thought to contribute to social complexity. Second, hyenas are super adaptable. Their geographic range in Africa is huge. This shaded red area, which shows their range, encompasses a super diverse array of habitats, from savanna to montane forest, and also includes fully urban ones. And I suspect that their adaptability might be related to behavioral flexibility. In order to measure behavioral flexibility in wild hyenas, I decided to test two specific skills, inhibitory control and innovation, because these are both important aspects of behavioral flexibility. Inhibitory control is the ability to resist a prepotent behavioral response in favor of a better one, which is especially important in complex societies where restraining impulsive behavior is crucial for maintaining social cohesion. Next, I also measured innovation, the ability to solve a novel problem, which is thought to be especially important in adapting to novel environmental conditions, such as those created by urbanization. I tested inhibitory control using the cylinder test, where I presented hyenas with a clear cylinder that rests horizontally on the ground. The cylinder is baited with a piece of meat in the middle, and the hyena has to inhibit going straight for the food and instead detour to the openings on either end. This subadult female here fails the test by bumping into both sides of the cylinder before retrieving the food. However, she did also have some successful trials, and you can see that here, where she looks through the wall of the cylinder, but successfully inhibits going straight for the food and instead detours to the opening on the side. And I tried to give every hyena at least 10 trials, and we scored hyenas based on the number of passes and failures. And I used a novel extractive foraging puzzle to measure innovation. This video shows hyenas solving this puzzle box. It has 4 different doors on each of the vertical faces, and each door required a different motor behavior to open and retrieve the meat from inside, as these hyenas are demonstrating for you. I tried to give every hyena repeated trials to see just how many of these 4 different doors they could open, and ultimately, hyenas got a score of 0 to 4 for the number of different doors that they solved. Hi everyone, I'm Lily. Based on the social intelligence hypothesis, I first predicted that hyenas living in larger groups would have greater inhibitory control, and I measured group size as the overall size of a hyena clan and as the size of a hyena's juvenile cohort, which counted the number of other cubs at the communal den, where unrelated females all keep their cubs, when the focal hyena lived there. And I found support for this prediction. Hyenas living in larger clans and larger cohorts had greater inhibitory control. So here we see clan size and cohort size on the x-axis, and the probability of passing a trial indicating successful inhibition, on the y-axis. And what we see is that both hyenas living in larger clans and those growing up in larger cohorts have a higher probability of success with the cylinder test. And this relationship was slightly stronger for cohort size. Next, hyena societies are also marked by these strict linear dominance hierarchies, and we assigned each hyena a numeric rank based on this hierarchy. So if group structure is also important for flexibility, the social intelligence hypothesis would predict that low-ranking hyenas should have greater inhibitory control because they must more frequently inhibit behaviors like feeding around high-ranking hyenas. And while I didn't find an effective rank alone, I did find an interaction between rank and clan size. Here we have social rank on the x-axis, and I scaled and centered social rank from -1 to 1, where the lowest ranking hyena in a clan was given a score of -1, and the highest ranking hyena in a clan was given a score of +1. On the y-axis is the effect of clan size on inhibition. So if we look at just low-ranking hyenas, we can see that the effect of clan size on inhibition is significant and positive. This means that low-ranking hyenas living in larger clans had greater inhibition than low-ranking hyenas in small clans. Next, if we look at just high-ranking hyenas, we can see that the effect of clan size on inhibition is not significantly different from 0. In other words, high-ranking hyenas' inhibitory control did not vary with clan size. Hi everyone. In sum, it seems like both the size and the structure of a hyena clan have important influences on inhibitory control, especially during development. This finding supports the social intelligence hypothesis and helps us understand the features that make a society complex. Next, in order to test the cognitive buffer hypothesis, I compared innovation with the puzzle box across 3 different populations of hyenas experiencing different levels of environmental change created by urbanization, because urbanization creates some of the most novel and variable habitats that exist today. The first population of hyenas lived in a very stable area in the center of a national park where human disturbance was very low. The second population lived on the border of a protected national park and overlapped a rapidly growing human settlement. This part of the park has experienced rapid change in human growth, buildings, tourism, and cattle grazing, and hyenas appear to be shifting their movement patterns and their diet to include more livestock while also scavenging from human trash. The third population was a fully urban population. These hyenas den in the churchyards on the outskirts of the city and travel in and around the city to scavenge from trash and livestock remains every night. Hyenas in both populations 1 and 3 have had many generations to adjust to their environments, and these environments should pose much less in the way of novel challenges for hyenas compared to the challenges posed by the rapid change that hyenas in population 2 are experiencing. Therefore, the cognitive buffer hypothesis would predict that hyenas in population 2 should have much higher levels of innovation than those in population 1 or population 3. What I found, however, was something completely different from my prediction. I found that hyenas in my rural population had significantly higher innovation rates than those in the transitional or urban environments. Furthermore, hyenas in the transitional and urban environments were not significantly different from each other. So what's going on here? Why are hyenas in the rural environment so innovative? Well, in carnivores, one of the largest correlates of brain size is hunting large prey, and for hyenas in the rural population, most of their food comes from hunting wild prey, while hyenas in the urban environment are mostly scavengers. And it may be that hunting wild ungulates requires a high level of innovativeness in hyenas. So, at least in terms of finding food, city life might actually be easier for hyenas and reduce demand for innovation. So, to wrap up, I found that group size and social rank both correlated with greater behavioral flexibility, which supports the social intelligence hypothesis. Second, hyenas living in the most stable environment had the highest innovation rates, which challenges the cognitive buffer hypothesis. Instead, it's possible that innovation rates correlate with hunting behavior, which could support either foraging or social hypotheses. And this is something I hope to investigate in future research. So with that, thanks for watching, and I look forward to your questions at the live Q&A session up next.